All right, let's go through cellular respiration and the steps involved in cellular respiration. The very beginning of cellular respiration starts with glycolysis. Glycolysis is got glyco, which means sugar, and lysis means rupture or break it apart. So this is our very first start at cellular respiration, taking that sugar and breaking it apart with glycolysis. Um, we are going to talk about how the products of cellular respiration are going to be different types of molecules, and one of those molecules is our energy currency molecule, the ATP, adenosine triphosphate. ATP is a nucleic acid. We saw nucleic acids before, and the nucleotides. Oh, God, sorry. The nucleotides consisted of a sugar. In this case, for ATP, it is ribose, which had both of those hydroxyl groups. It had the nitrogenous base of adenine, your A, and adenine always bind with thymine if we were talking about DNA, and adenine would bind with uracil if we were talking about RNA. In this case, we are just talking about ATP. It's its own lone molecule is ATP. It has adenine and ribose to combine makes adenosine, and then it is triphosphate, which means it has three phosphate groups. So we have alpha, beta, and gamma, one, two, three different phosphates here. And our phosphates have negative charges. So each one of these has a negative charge. And if any of us remember, from like kindergarten, preschool days about magnets, and if you had two magnets together and you had two negatives, they wouldn't want to go together, right? If you flipped it and you had a negative and a positive, then they would stick together. But two negatives kind of, they don't attract each other. They actually repel each other. So these have slightly negative charges. So that's kind of important because we said that this is adenosine ATP. ATP is our energy currency molecule because it acts like a rechargeable battery. This is the third phosphate, this gamma phosphate group over here. And this one snaps on and off. It breaks off, and if we break a bond, we release energy, right? And it will go from ATP to ADP, adenosine diphosphate. And then it has this lone little phosphate over there that is broken off, and it will need to be attached, and then it will come back later when we receive chemical energy, like we talked about in creating these ATP molecules. Later, when we receive chemical energy because we just ate dinner or had a smoothie, milkshake, or whatever, we're going to take that ADP, tack on that third phosphate using the energy in our chemical reactions, and now we have recreated ATP. So we have this cycle going back and forth between ATP, busting it off, releasing energy, energy goes out, ADP in its third phosphate, and then when we tack that thing back on, energy has to come in, in, energy comes in to tack on the third phosphate and bring it back to ATP. Energy out. So it's a rechargeable battery, ATP, ADP, ATP, ADP, all day, every day, 24/7. Metabolism is going. So this is ATP, our energy currency molecule. Glycolysis. Step number one in cellular respiration. Cellular respiration is complicated. It can be a bit overwhelming. I'm going to break it down into three basic steps. On these three basic steps, I'm going to want you to know some information for every step. Let's write down the information. So I want you to know what location is this happening? Where is glycolysis happening? Where's glycolysis happening? This is happening all over the cell in the cytoplasm. Happening in the cytoplasm. This is where this is happening. Glycolysis is going to be located in the cytoplasm. And is it aerobic or anaerobic? Does it need oxygen or not? That's another thing that I'm going to want you to know for every single step. Glycolysis is anaerobic, meaning it does not require oxygen. So no oxygen needed for this step. So know where is happening. Does it need oxygen? No, it doesn't. And then what are its products or results at the end? So with glycolysis, we're going to take in one molecule of glucose. And if we remember glucose, we wrote it as C6H12. H12O6. And we're going to take that molecule of glucose that has 6 carbons as its backbone, and we are going to split it in half. We need to get this reaction going. Remember, we said that nothing happens without an activation energy. It's not just going to turn on and this is going to happen. So to get this activated, we're going to bring in 2 ATPs. We have to get 2 ATPs to get the ball rolling on glycolysis. So our 6 carbon molecule is now going to split. I'm not going to ask you about any of the intermediates. Excuse me. There are a lot of intermediates in this process. I don't want you to know them. I don't want you to know any of that, but I want you to know what it ends up producing at the very, very end of all the steps. At the end of all the steps, we start off with glucose, C6H12O6. We end up with pyruvate. Pyruvate, this 3 carbon molecule is what we end up with, and we end up with 2 of them. So we took 6, we got our 6 carbon molecule, this is our glucose, and we end up splitting it, lysing it in half into 3 carbon molecules of pyruvate. So here's a pyruvate, and here's a pyruvate that came from 1 molecule of glucose. So we split it, we lyse it in half. Glycolysis is splitting glucose in half. At the end, it's going to end up with 2 3 carbon molecules of a substance called pyruvate. So what do you need to know from glycolysis? Where is it happening? Again, cytoplasm. It's happening in the cytoplasm of the cell. Every cell does this. Eukaryotes, bacteria, everybody does cellular respiration. This is happening in the cytoplasm. Does it need oxygen? No. It is anaerobic. This is an anaerobic step. This is step number 1 is glycolysis. Happens in the cytoplasm. It's an anaerobic process. At the end, we're going to produce 2 pyruvates. I do want you to keep track of the number of ATPs. That's one of the few things I'm going to ask you to keep track of on each and every step. So 2 ATPs went in, and we end up producing a total of 2 here and 2 here. So we produce a total of 4. If it cost us, if we gained 4 ATPs, we gained 4, I can count. We gained 4 ATPs, but it cost us 2 to even start off the reaction. We only end up with netting 2. So let's say that again. We produced 4. Yay, we made 4 ATPs, but it cost us 2 to get the whole ball rolling on this process. So we end up netting 2. We're going to net 2 ATPs. No. Sorry, the dog. So those are our results. Those are our products from glycolysis, which is step number 1 out of the 3-step process of cellular respiration. Again, glycolysis is breaking up glucose, a 6 carbon molecule, into 2 3 carbon pyruvates at the end. To do this process, it's happening in the cytoplasm. It is an anaerobic process. We end up with 2 pyruvates, and we end up with those 2 ATPs at the end. Those are our takeaways that I want you to know for glycolysis, step number 1 in cellular respiration. We're going to move on now. So at the end, we had 2 pyruvates. Let's hold on to those pyruvates, and we're going to decide, what do we do with those pyruvates? If we have oxygen, we are going to take those pyruvates and move on to the citric acid cycle. Here is our glucose molecule up at this picture. There's our ATP coming in. No, go back. And at the end are 2 pyruvate molecules, 2 pyruvates, and a net of 2 ATP at the end of this step. And if we have oxygen, we're going to continue on to the next step of cellular respiration, which is going to be the citric acid cycle in the mitochondria. When you see these steps, glycolysis, citric acid cycle, you're going to see some energy carrying molecules. Those energy carrying molecules, let's see if we can find one. Here's one, NADH. NADH, I want to talk about these just real briefly. I'm not going to ask you to count these up, but I do want you to understand the concept of what's happening when you see these molecules. So NADH, we have an NAD right here, and then you can see it changes to NADH. It gained a hydrogen. It went from NAD to NADH. It gained a hydrogen. And if we remember from chapter 2, every atom, so a hydrogen atom, is going to have a proton and an electron, right? As long as it is a neutral atom, and it should be neutral, it will have one proton and one electron, and maybe it has two protons and two electrons, but it will have a balance between protons and electrons. And in this case, it's hydrogen, so it just has one proton and one electron in this situation. So NAD plus its hydrogen, NADH, it gained a hydrogen, it gained a proton and an electron to become NADH. So I'm telling you that because we keep track of electrons. That NAD molecule is an electron shuttler molecule. That's what it's going to do. NADs and FADs are going to shuttle electrons. So when we're talking about gaining or losing electrons, you may hear the terms oxidized or reduced So oxidation, to remember if something is getting um, gaining or losing an electron. Oxidation is going to be a loss of electrons. Oxidation is loss. Oil, reduction, if something is reduced, reduction is gain of electrons. So these reactions are called redox reactions, reductions and oxidations combined. Anytime something is reduced, something else is being oxidized. Those electrons are being shuttled back and forth. So let's look at this one more time. We see an NADH, and it is going to gain a hydrogen. So if it gains something, was it oxidized or was it reduced? Let's remember our oil rig. Oxidation is loss, reduction is gain. So it gained a hydrogen, which means it gained electrons. So NADH is reduced. NAD becomes reduced to NADH. It gained electrons in the form of that hydrogen. So these NADs and FADs, they shuttle electrons back and forth. And those electrons are going to be important when we get to the final step of cellular respiration with the electron transport chain. Those electrons will be important then. So I don't want you to calculate how many NADs or FADs. I'm not going to ask you to keep track of that. I do want you to keep track of ATPs, and I want you to understand what NADs and FADs do. They will shuttle electrons. They will either gain electrons or lose electrons. They will gain an H or they will lose an H because they will be oxidized or reduced when they do that. So we just covered glycolysis. Glycolysis was an anaerobic process. It happened in the cytoplasm, and our end products were a net of 2 ATPs and 2 pyruvates. And if the pyruvate is in an environment where oxygen is present, then we are going to move on to the citric acid cycle. Where is this happening? Citric acid cycle is numero dos. It is step number 2 of cellular respiration, and this is happening in the mitochondria, but not just anywhere in the mitochondria. It's happening in the matrix. If we remember from chapter 3, the mitochondria looked like, I called it a jelly belly. It was kind of round, it had double membranes, and these membranes folded in. All these membranes that folded in. The membranes that folded in were called cristae, and the fluidy part in between these membranes was the matrix. So this is happening in the matrix of the mitochondria. So need to know where it's happening. Mitochondrial matrix. Is it aerobic or anaerobic? This process is aerobic. It has to have oxygen or we don't go to the mitochondria and we undergo fermentation, which will be later on. So this is aerobic. We need oxygen. Okay, now we said that the step before this, glycolysis, produced 2 ATPs, which is great, and it produced 2 pyruvates. What are we going to do with those pyruvates? That's this step here. So we're going to take those pyruvates, and this little step here is technically called the intermediate step. It is going to change pyruvate and we're going to turn it into, we're going to see, here's our those electron molecules again, NADs and NADHs, and we are going to tack on a coenzyme. And so we're going to change pyruvates into a molecule, and that molecule is called acetyl-CoA. Acetyl-CoA. Acetyl-CoA feeds into the citric acid cycle. So this is our beginning molecule, acetyl-CoA. And acetyl-CoA is going to go through this cycle. There's going to be lots of intermediates in the citric acid cycle. I'm not going to ask you to know those intermediates either. You don't need to know them. I do want you to know that this is happening in the mitochondrial matrix. It is an aerobic process. It takes pyruvic acid, changes it into acetyl-CoA, and that acetyl-CoA goes around and around and around and around the cycle. What do we end up producing at the end of this cycle? I want you to know that we gain another 2 ATPs at the end of the cycle, and that this is where CO2 gets produced. CO2 is going to get produced in this cycle, our citric acid cycle. That CO2 that we exhale when we end up with cellular respiration. Oh, just think about it. Oh my goodness. Just thinking about exhaling makes me want to yawn. And I am exhaling out CO2 when I do it. That is happening at step 2, citric acid cycle. So we're going to go over this one more time. Step 2, it is going to take pyruvate from our previous glycolysis. We have oxygen present in the environment, so that pyruvate is going to be changed in something called an intermediate step to acetyl-CoA. Acetyl-CoA runs through this cycle called the citric acid cycle. It's called the citric acid cycle because one of those guys is citrate, and that gives it the name. You may also hear it called Krebs cycle. Krebs with a K, Krebs. Krebs cycle is the same thing, just called a different name. As it runs through the citric acid cycle, this whole circle where it runs through a whole bunch of different intermediates and then eventually runs right back to where it started at, that's why it's called a cycle. It's going to produce some things. It's going to produce those energy carrying molecules, NADHs. Don't memorize how many, just know that it makes them. It's going to produce some electron carrier molecules, FADHs. Don't memorize how many, just know that they make them. I do want you to know that it produces CO2, and I do definitely want you to know 2 more ATP got produced. So far we produced 2 ATP in glycolysis, and we have produced 2 more ATP in citric acid cycle. So we're up to a total of 4 in this citric acid cycle, and it's producing 2 for every pyruvate that comes in. We are going to produce 2 ATPs. So let's move on. We are now going to stay in the mitochondria. Mitochondria, got the mito, but we are moving from the matrix, the liquidy part, into the membrane. So this phospholipid bilayer right here, this is the membrane. So this is going to be the mitochondria cristae is where this is happening. So this process where, and it is definitely aerobic. Yes, we need oxygen for this. Remember all those NADs and FADHs that I told you not to worry about counting, but just understand why they had those molecules and they gained all these hydrogens. We had all these all these NADHs, all these FADHs. They had gained these hydrogens, they had gained these electrons. This is their time to shine. It is this step. Step number 3, the third and final step of cellular respiration is the electron transport system. You may see it called electron transport chain, ETC or ETS. And those electrons, all of those H's, all those hydrogens that were stuck on those electron shuttle molecules, they're important now. We are going to use them now. So this third step is where they get their moment in the sun. So here's one of the many NADHs that got produced. And look at it, it's going to split. The hydrogen's going to come off. We're going to end up with NAD. And here's our hydrogen up here. And look, hydrogen has a positive. What ion was it that had a positive ones? Cations or anions? Cations. I'm telling y'all this because this is all going to be on the midterm. And even though, this specifically is not asking you about it, This is all stuff that's building on each other. So y'all be ready for the midterm when you see this. So an NADH has lost its hydrogen. It has been, if it lost an electron, is it reduced or oxidized if it lost an electron? Let's remember back to oil rig, oxidation is loss and reduction is gain. So if NADH lost its hydrogen, it's oxidized. NADH got oxidized. Its hydrogen is now up here. It got shuttled through these little proteins. So here's this little purple, it's not purple, it's bluish, isn't it? It's blue protein here that has pulled and taken away and shuttled away the hydrogen off of the NADH. So NADH got oxidized. It's now NAD, and the hydrogen up here is here, and it's hydrogen and it's a plus, which means its electrons split off. This is a cation. The hydrogen up here is basically just a proton. It's a proton. Hydrogen up here. And the electrons are floating through these blue proteins. These are called quinone proteins. And so they basically play hot potato with the electron. The first guy pulls off the electron, you take it. No, you take it. No, you take it. You take it. You take it. You take it. And they shuttle it. So it's the electron transport system. The electrons are literally being transported through the membrane, through these quinone proteins. And so you take it, you take it, you take it, you take it. I don't want it. And eventually they're going to end up over here. We're going to end up over here and we end up with this pump. This is guy over here is called the ATP synthase pump. And so the job of the ATP synthase is to make more ATP at the end. And what it does is it's going to use all these protons up here, all these hydrogens. So we said that we made a whole bunch of NADHs and FADHs, and we are pulling off the hydrogens, and the hydrogens come up here. All of these hydrogens are up here. Hydrogens with their little protons are up here. Hydrogens. And so we have this really high concentration of hydrogens up here, and the electrons were going through the proteins. The electrons were shoving through the proteins. They are not up there. Well, if we remember back from, I want to say chapter 2, we talked about diffusion, right? And it was passive, and diffusion is always going from an area of high concentration to an area of low concentration. Everything always wants to go to an area of lower concentration. If you have somebody, if you're somebody's getting ready in the morning, right, and they are getting ready, they're putting on all their product, doing their hairspray, or maybe they're wearing cologne or something, and you smell that, the smells from those things will waft out of the bathroom and into the surrounding room because that high concentration is going from an area of high to low, and you will smell that smell as it travels from high to low. Same thing with the hydrogens. The hydrogens want to go from high to low. So those hydrogens really want to get inside on the other side of this membrane. And since they want to do that, this pump's going to take advantage of it. The ATP synthase pump takes advantage of that difference between the high concentration of hydrogens on the outside of the membrane and the low one on the inside. So the hydrogens come through. And here we've got a hydrogen. It's going to come through. And as it comes through, it powers this pump. And it's going to power the pump, and the pump is going to use that energy as the hydrogens come through to snap on that third phosphate, to tack it on. So we're going to go from ADP and its lonely little third phosphate that had broken off, and we're going to use the energy from this pump to now create ATP. So we said, ADP was our rechargeable battery, and we went from ATP, broke off the third phosphate, used that energy, and we ended up with ADP. Now is the time to make that back up. So we're going to have ADP turn into ATP with the help of the ATP synthase pump, using all those hydrogens from those electron shuttle molecules to power this pump. So we end up producing ATP, and then those electrons, we can't forget about the electrons that were playing hot potato, like sad little loser electrons being pushed along through the membrane. We're going to use them. They're not losers. We totally want them. So we are going to use those, and when those hydrogens are over here, we're going to have all these hydrogens, and they're going to come through, right? They're going to bind with the oxygen because we said this was an aerobic process. We need oxygen. So we have all these hydrogens, they're coming along, and we're going to bind them with the oxygen. And in the end, we're going to produce H2O using the electrons, snapping them back on with their hydrogens and the oxygen. And we're going to create H2O when we do that. So our byproducts at the end of cellular respiration is going to be water. We're going to produce the water, and we're going to produce, remember our previous step one was only 2 ATPs, and glycolysis was 2 ATPs, citric acid cycle was 2 ATPs. Look at the number of ATPs you get from this step. This is the most efficient step, is the electron transport chain, electron transport system, 32. You get 32 ATPs on this step. So this is the coolest step, is this electron transport chain, electron transport system being run through with those NADHs and the FADHs. Their hydrogens all come up to the top. The hydrogens then power this pump that cranks out 32 ATPs and also makes water molecules at the end. So we end up with C6H12O6 plus 6 molecules of oxygen will give us, when we break it down, CO2 and water, 6 into 6. Plus ATP. And then when we do this, remember we said that energy could not be created or destroyed, but it can be transferred. So when we broke apart this glucose molecule and we busted it up and we ended up producing energy because we broke those bonds, we harvested that energy in a total of 32 plus the 2 ATP from glycolysis plus the 2 ATP from citric acid cycle gives us a total of 36 ATP that we can produce from one molecule of glucose. So we produce 36 ATPs, and then the other bit of energy does not all get contained in that ATP. So the rest of it's going to go off as heat. So when we break down glucose, 40% of it is harvested into ATP, and 60% of it is energy is lost in the form of heat, in the form of heat. So just by breaking down, chemically breaking down glucose and performing this process of cellular respiration, we end up producing heat, and we can capture 40% of that energy in the form of ATP. How much ATP? You need to know, and I need you to know. 2 ATP in glycolysis, 2 ATP in the citric acid cycle, and a whopping 32 ATP in the electron transport chain, electron transport system. You may also hear it called oxidative phosphorylation. That is this third step. So 36 ATP, depending on which textbook you're reading. Some of them say 34, some of them say 36, but that is the total amount of ATP that you are getting from a single molecule of glucose and 6 molecules of oxygen. That's how much you're producing. So I need you to know every step, know the 3 steps, know the things that I want you to take away from them. Which step called glycolysis, citric acid cycle, electron transport chain? Where is that step happening? Glycolysis was happening in the cytoplasm. Citric acid cycle is happening in the mitochondria, in the matrix of the mitochondria, and electron transport chain is happening in the cristae of the mitochondria. Is it aerobic or not? Glycolysis, remember, was anaerobic. It didn't need oxygen to perform that process. And citric acid cycle needed oxygen. Electron transport chain needs oxygen. Those are both aerobic. Then what are the end products? At the end of glycolysis, you had 2 ATPs and you had 2 pyruvates. A citric acid cycle was going to take that pyruvate, change it to acetyl-CoA, and then acetyl-CoA will run through the entire citric acid cycle, and you end up producing CO2, some NADs, some FADs, and 2 ATPs. So now we've got a total of 4 ATPs. Then we're going to come on down, and we can run through the electron transport chain, the electron transport system, oxidative phosphorylation, and get 32 ATPs at that step for a grand total of 36 ATPs. So super cool. I need you to know the 3 steps. I need you to know the name of the step, where the step is happening, what are the products, of that step, and was that step aerobic or anaerobic? Those are your four big things I need you to know for every single step involved in cellular respiration. I want you to be able to recognize the steps, the equation for cellular respiration. Be able to recognize it. I need you to know the breakdown of energy. That when we broke down that glucose molecule, that 40% of it does get harvested as ATP. We can use that energy to convert ADP to ATP. 40% of that energy goes there. The other 60% of that energy is transformed and changed into heat. Changes into heat. All right, so I'm going to cut this video here and then we'll make a new video on the last two sections.